Okay, I'll get started. Welcome and thanks for joining us for the third in our uh, webinar in our 3Rs series for the fall. Today's webinar, Microphysiological System Basics and Application in Early Preclinical Development, is being presented today by Dr. Norman Peterson. Let me briefly introduce Dr. Peterson before we begin. Uh, Dr. Peterson's expertise does span preclinical pharmaceutical development and the advancement of supporting technologies. His research and collaboration focus on the development of NAMs with an emphasis on investigative toxicology, microphysiological systems, and non-invasive in vivo imaging. He actively participates and serves as a leader in a number of different consortiums, including our co-hosts for the series and our 3Rs conference and related content, the 3Rs Collaborative. So thank you for being involved in so many groups, Dr. Peterson. And Dr. Peterson's education, training, and experience includes time at the University of Illinois, Wake Forest, University of Pennsylvania, and Johns Hopkins, as well as multiple life science companies. So we're very pleased that Dr. Peterson is sharing his expertise with all of us today. And this is a longer version of a presentation that Dr. Peterson shared with our 3Rs conference last spring. So it was well received, and we're glad to be able to share an expanded version of that and provide some additional details and research for everyone. So with that, bear with me, and I'll turn the control over to Dr. Peterson.
Well, thanks for that introduction, Tom, and I appreciate everybody attending this webinar. Lately, there's been a whole slew of microphysiological systems webinars, so I appreciate your tuning in to this one. Before I get started, let's kind of, a lot of topics we'll be going over, and we'll be jumping around a bit. So this is kind of what the field looks like. I'm going to give a brief background in addition to what Tom did, just to provide context of where some of my comments and perspectives are coming. We'll talk about the hype that's been generated by the FDA announcements. And I think to understand the microphysiological systems, we have to put that into the perspective of animal models in drug development. I know several of the attendees are from industry, so if some of this is redundant, hopefully it'll be put in a different type of perspective and still be interested. And then we'll talk, then eventually we'll get to the microphysiological systems, the FDA perspectives, current applications, and then I'm going to offer some of my opinions. So to start off with, Tom went over this sort of in the introduction, and this is kind of all the places where I've been. The V is my veterinary school, A is academic institutions, my early career, and then B is biotech pharma, and most recently I'm up in the upper left for the last 5 years, enjoying the Pacific Northwest. I've been a clinical or attending veterinarian at each of these places. And I've worked with developing several different models, animal models. Mainly, I've been in the departments of toxicology, so that's helped my understanding there as well. And also, as I started my biotech career, I've developed some in vivo imaging cores. I'm a big advocate of using environmental health monitoring. And as was mentioned previously, I've been involved with a variety of different 3Rs organizations, and 3Rs is kind of at the core of my research. So that's my animal experience. I've also had some tissue experience. And the top 5 or even it predates what we call microphysiological systems. I think these are sort of the precursors to what led to the development of the microphysiological systems. And then the bottom 3 are more recent developments that I've either collaborated with or have been projects in my lab. And we'll touch upon some of those a little bit later. So why am I here? Why am I giving this presentation? Well, the FDA recently announced that we're going to phase out animal testing. And that has generated a lot of confusion and discussions and all. But on the flip side, in my early career when I was in academia, I submitted several grants trying to convince the government to look at the three R's. We need to do more research in this area. And talking with others, it was really frustrating. Government kind of dismissed us, sometimes even put us in special interest groups, and it was very difficult. So I think with this announcement, we're really flying high. Finally, the government has looked and paid attention that the three R's are important, and hopefully that concept will stretch over into areas of refinement and other types of replacements. But one thing, when they make an announcement like this, it created a whole lot of buzz. And I've created here sort of a collage of things that I've pulled off the internet and various website pages and all, and even LinkedIn. A lot of discussions on this area and where's FDA going, a lot of confusion. So I'm hoping that with this presentation, we'll put things into context and we can have a better understanding of our current situation. So with that being said, let's ground our balloon, look at what needs to be done. It's great that we've got the funding, but now the work gets done, and let's take a realistic look at where we're at. And to do that, I'm going to rely on this little diagram here that I picked up doing an image search, and I think it really well depicts on how we go about solving problems and eventually getting to success. So I'm going to be referring to this diagram throughout the presentation. So first, let's start off and define what the problem really is. And so it's been mentioned that animal models are not predictive. And to some extent, that's true. We know when we take drugs through the development process, it's somewhere around like 15% that are only successful. So there's room for improvement. But should we really be throwing out the animal models if they're not predictive? Let's take a closer look at that. So this is a classic schematic of how drugs are developed. And again, my friends in industry who have seen this little chart ad nauseum, but it describes how pharma biotech takes drugs and develops them to meet FDA requirements. So if you start over toward the left, it's where somebody gets an idea that, hey, this might be an interesting target, or this might be an interesting way that we can attack a certain disease. It's largely in vitro. They do a lot of literature searches, and then they take cell systems. We're mainly looking at the mechanism to see if we can interrupt some disease pathology. So for the most part, that early study is in vitro. And if you've got really robust and high quality in vitro systems, that in itself is a reduction, because you're building a case before you even put it into the animals that this is going to work. Then the preclinical development phase is more where the in vivo aspects come in, the testings. And what we're trying to do is set this up and do enough studies to convince the FDA that we can put this compound or this chemical into humans. The clinical development is divided up into three different phases. We'll talk a little bit more mainly about the first phase. This is where it goes into healthy humans, just to make sure it's safe. I might point out that the FDA pretty much gets involved in pretty much the later phases of preclinical development. This is where discussions occurred that the strategy is proposed, and discussions begin to get in place that to meet the FDA requirements, and then the FDA follows the project along throughout clinical development. So that, in a nutshell, is how drugs are developed. But let's look a little bit closer at that preclinical development phase that involves mostly the in vivo, some in vitro aspects, but we're focusing on the in vivo aspects here. And I define sort of the three pillars that we need to get in order for the FDA. Is one, we have to show that the drug is efficacious, that it does something, that it does what we claim it to do. So there are a certain set of studies that are done depending upon the disease and the models that we have. And the second pillar is the safety and toxicity. And this is very important. We don't want to put anything in a human or an animal for that matter that will cause severe adverse effects. The drug metabolism and pharmacokinetics both impinge upon the efficacy and the safety. The drug has to be in the system long enough to have an efficacious effect, but sometimes if it's in there too long, it can have a toxic effect. And this relationship between efficacy and safety is called the therapeutic index. I'm going to take a little bit of time just to describe this because we're going to come back to it. So let's take you've got a drug of choice, and we tested it at various levels because we want to find at what dose it's toxic. There's a saying that everything is toxic. It's all a matter of dose. I mean, you give an animal or person too much water, that could kill it. So it's all about the dose. So let's start out with our drug that we're looking at, and we do our efficacy studies. And let's say it takes a very small amount of drug to cause that effect. Let's say 5 milligrams, for example. And we keep dosing in our animal system or in vitro system, and we get up to 500 milligrams that show that it's toxic. So you notice there's a big space between this. So if we test this in animals, we've got a pretty low risk that it's not going to cause harm to the animal. At least we're not saying this for sure. We're evaluating risk here. But on the other hand, let's say that we put our drug in there and it takes about 50 milligrams to cause an effective result. But we can only go up to maybe 70 milligrams before we start seeing toxicity. So that only gives us a space of about 20 milligrams. The therapeutic index is going to be a high risk to use this, and probably that drug may not go through. So therapeutic index is the hallmark of drug development. So we'll come back to that this diagram a bit later. So let's talk about the efficacy point of that. And I'm going to use cancer because I've done probably 70% of my research in the field of cancer biology. And what we do is we give human tumors to immunocompromised mice, and then we treat them with our drug candidates and see what happens. Now, if that drug doesn't shrink, and we test this in let's say 3 mouse models, that we have a high level of confidence and it doesn't work, that's pretty much a showstopper. We won't take this further unless there's other evidence. But I haven't seen a drug go forward that really hasn't worked well in an animal model. And I think this is probably pretty highly predictive, just based upon the relationship of the physiology of the human and the mouse. Maybe we are throwing out a few compounds, but for the most part, I think this would have a high predictive value. We don't know because we never did stick it into a human. I think it wouldn't make sense to do this if it didn't work in a mouse model. Now, on the flip side, let's say that the drug shrinks, then we might have a good case for taking it forward. However, we need to keep in mind if we consider these tests, we've cured cancer over and over again, but we've cured it in the mouse. So a lot of these that have gone forward have a low predictive value. A few do make it through, and it is translational. So I think what I'm trying to emphasize here is that the animal models do have really value in what we're doing, and they're still needed in our studies. Let's look at the other end of the therapeutic index, and that's the toxicity. And in working with toxicologists and just sort of a over umbrella type viewpoint, it's pretty much a fishing expedition, especially if you don't know anything about the compound that you're developing. So what they do is they take various doses of a drug and put them into the animals and ask the pathologists and the people that are doing the in vivo studies, does this make the animal sick? We don't know what organ system it might affect, but this is why we do it, to understand more where it's affecting it. So in this realm, the animal model forms a sort of a screening mechanism that we use. This looks a little bit complicated, but let's take a look at sort of this. I think it's called a chi-square diagram here. Let's focus over on the left-hand side, and this is sort of a, let's take a rat study for example, and focus on that. And so if we put our compound in the rat, we can find that it has a very narrow therapeutic index based upon the efficacy studies and toxicity, or it could have a high therapeutic index. Now, if it has a very low index, we're not going to put this into an animal. And this is what I call our never data. We'll never get that. And I don't know anybody that's critical of the animal models that would say, yeah, it caused sudden death in the animal. I'm going to go ahead and try it in my clinical trial. That doesn't happen. That's data that we never get, but it's very valuable in determining what the toxicity is. On the other aspect, though, let's say that the rat tolerates it quite well. If we put it into a human in phase 1 studies, and we've done due diligence with our animal and in vitro models, it's very rare that in phase 1 we're going to have an adverse effect. So that's illustrated by the lower right-hand square. And then the part that's really important is if it looks well in the animal, is it tolerable in the human? And there's some variability there. This is kind of where I think the box where we need the most improvement. I'm going to talk a little bit about this bottom right square here. Oh, I forgot to mention too, it's sort of we're using the animals as a canary in the coal mine. And if the canary dies, the person isn't going to take off his gas mask, and that's what we're doing in those top columns or top rows. So there have been a few clinical disasters, and people that advocate that the animals are not predictive. They use the T. Gennaro study over and over again. This happened in 2006. 6 patients had a cytokine storm that really the animal models did not predict. And then again, I'm aware that there was an incident in 2025. This was actually put into some pediatric patients that had Duchenne's muscular dystrophy that caused some death. But over a period of approximately 20 years, it's a rare occurrence. And I think that's because we have things in place, procedures, or animal testings, and the way that we run clinical trials that we avoid these sudden deaths. And that's one of the main things that we're trying to do with our toxicity studies and our animal models at this point in time. And this was just another study that was followed up from the T. Gennaro, showing that over a period of 4 years, there were no deaths, and the number of mild to moderate disease incidences was quite low. Again, just attesting to the value that we're currently using with animal models. So instead of calling this sort of a problem, I think it's better to define it as a challenge. It's always healthy to look at problems in this way. And most of the toxicologists that I've worked with are excited about meeting this challenge. Can we do it better? And so to define really what the challenge is we want to look at ways that we can develop drugs that are predictive or even more predictive than our animals without using the animal models. One challenge that we do have is we're not quite clear on the metrics of how well our studies work right now. And that's especially when we get into phase 2 and phase 3, we start looking at patients. Some of these are already compromised in the area of cancer. They've been exposed to a variety of chemotherapeutics. So it's very hard to determine, at least from a toxicology standpoint, how predictive the models are. And so it's one of the things just to keep in mind. So there is a proposal for a possible solution. So let's take our little guy and see if we can step up to this next platform. And here we get into what is a microphysiological systems, and there are a variety of definitions. And usually it will consist of two or more of these components. It's a multicellular environment, so as opposed to the tissue cultures that were working in the past, where we take a single cell line and put it and expose it to plastic, um, this is different. It uses at least two different cell types, maybe a supporting cell type, maybe blood vascular cells, and they grow usually in a matrix. Matrigel is commonly used, but other groups are looking at different types of things. The cells, because they're grown in this way, can form their natural 3D structure, or they're layered to represent what it would look like in the tissues. And what is neat about some of these systems too is that they're dynamic. They try to represent the blood flow that we see in vivo. And there are even some examples where stretching to represent lung breathing and peristalsis that are incorporated in that. I've got a few examples of those. And we use either primary stem cells that we isolate from people or humans, or stem cells that we treat with different media to form different lineages. And I copied this from a recent publication. I think it pretty well illustrates sort of the basic components of microphysiological systems. And usually the chips, sometimes it's called tissue on a chips. A lot of times those are bioprinted. They tend to be some type of a plastic. They form the framework. And then most of these systems have some type of a flow that's governed by a peristaltic pump or some type to regulate the flow, mimic blood flow. Of course, you don't want your media blasting through there to cause shear stress on your system. So you've got a flow that goes through there. And then if you look over more toward the top right panel, you can see some of these pipes with endothelial cells to represent the blood system. And what's neat about these endothelial cells, they do form tubes when you put them in vitro. And in this particular study, they actually took some kidney organoid cells. And what they wanted to demonstrate is that from this major blood vessel, that the endothelial cells would then migrate up to the organoids and infiltrate them, much as you would see in a kidney. And they demonstrated that through these microvessels, using fluorescent dyes, that the organoids were permeated by these cells. So that's one type of organ on a chip. The other one that I find is really fascinating, coming back to that concept of stretch, is a lung on a chip. And if you think about it, there are basically two components that your lung tissue is exposed to. So it's air on one side, blood flow on the other. And this system tries to mimic that. And if you look at diagram number A, you can see the endothelial cells forming the vasculature. And this is where your media would flow through. And your media, the nutrients in your media, make it through that membrane. It's a semi-permeable membrane to feed the epithelial cells of the lung. Note that on the top, you've got an area of air. So you've got to balance this so that the cells don't dry out, but yet they have exposure to the air, much like you would have in an in vivo system. We took this a model one step further, and if you look at the panel in the middle panel in the over in the right, you see a vacuum. This is where a machine can apply a vacuum off and on, and that membrane stretches. So now your whole system is exposed to something that's much like a lung. Your cells experience the contraction and expansion that a lung would normally do. And they characterized some of these epithelial cells, and they found that the protein expression and the phenotype of these cells was much more similar to that what we see in vivo than in a stagnant culture system. So this is a lung on a chip example. Those were just a few, but there are many MPS different types of platforms that meet different needs. And I've listed some of the companies. There are 30 of some. The number is probably 4 times, maybe more than that. And you can actually search some of these companies. If you go to the 3RC website, they have a great system that you can go through and look for your organ of choice there. So I encourage you to take a time, just fish around at that site. So then to summarize, let's look at what are the advantages of the MPS system. So I think one of the greatest advantages, they're human cells. When we use animal models, of course, they're not human cells. And there are some differences in the physiology and the way that we metabolize drugs that are different. And so with the MPS systems, you can use human cells. One of the advantages also is it represents the human heterogeneity. Our models may not have that. That can be a plus or minus. The plus side is we want to look at a heterogeneous population, especially if we're looking at toxicity. The minus side is with heterogeneity, you increase your variability. So plus and minus there. Talk a little bit later. We can compare species. Not only do we have to restrict it to humans, we can do other species if we want to do those comparisons. Coming back, the mixed culture, the crosstalk among the supporting cells, the vasculature is so important, and it changes gene expression and the phenotype of these animals. So that having the neighbors of these cells to communicate with is so important. And we talked about mimicking the microenvironment, fluidics as well. The other thing is too, we're simplifying the system. We're taking it out of context. In most cases, you can't put in all of the cells that are involved in an in vivo system, so you can ask mechanistic types of questions without getting interference from the whole animal physiology. So those are some of the advantages. And with these advantages, the FDA has come out and said, we can use these, and what we're going to do is we're going to encourage the use of these to phase out animal testing. We're going to replace animal testing in the years to come. But what's interesting, if you go to another page where they define their roadmap, they talk about reducing animal testing. So there's a little bit of a difference here. I mean, I think of replacement as the ultimate reduction, but I think the timeline's a little bit different when you're talking about reducing versus completely replacing. So we're going to look a little bit closer. Are we going to reduce or are we going to replace animals? Let's take a look at this. But also, if you go to another FDA page, and I think it's important that the FDA, to keep in mind, they're given the responsibility that they have to assure the public that the drugs that are going in there are safe. This is what it's all about. And they're going to use science-based information to get at this. So what I'm getting at is if we do switch systems, we got to make sure that it's at least just as safe and that we're using good science to compare the two systems. So that was the outward-facing FDA proclamation. There's another group of FDA that I kind of think of is in the trenches, and that's the review team. They're the ones that interact with the biotech and pharmacy companies. They're the ones that give the go, no-go type decisions. They review all the data. They ask the questions. And this review team has the responsibility that the data is sufficient, that it's safe, and that the risks outweigh at least the risk for the benefits of what you're doing outweigh the toxicities. So the review teams are held responsible for this. So, the FDA may make certain proclamations, but when it comes down to it, it's the review teams that really where the buck stops. And they need to make sure that the data that you're providing, be it in vivo or in vitro, is sufficient. So we're ready to kind of move on, but we're going to do that using science and appropriate risk assessment as we move forward. And the public expects it, our scientists want it, and those reviewers also emphasize it as well. So let's take a step up and look at the solutions and what might be the barriers of using microphysiological systems before we can actually define that we have success. And one thing that most talks that I've gone to really don't consider these concepts in mind. I'll go through this quickly because it's pretty much basic biology. But we know the human nervous system that goes from the brain to the nerves feeds into all of these different organs. And we know that the nervous system has a profound effect on disease progression. There isn't an MPS system around that has these tissues with an intact nervous system that goes back to the central nervous system. So it's one deficit. The other thing that we need to keep in mind is the endocrine system. We know that our cortisol levels vary from day to day. MPS systems really can't mimic this, or they can't mimic when the animal is stressed and cortisol levels and other types of hormones go up and down. Bone marrow cell recruitment. So we know that when we have an infection or an insult to the body, there's cytokines that make it back to the bone marrow and say, hey, I need a certain type of cell type here. Let's start producing those. Those respond and go to the various tissues. That is very difficult to establish and hasn't been established yet. The other system is the microbiome. We do have in vitro systems that look at intestinal MPS, and they've added bacteria to that. And they can look at sort of the localized effects and see how they affect different systems, different types of cells within the intestines. But for those distal effects on how the microbiome might alter responses in the brain or lungs or other organs, haven't quite gotten there yet. So, in addition to all of these systems, can come together and have an effect, and we can't really replicate them and the interactions with them in a microphysiological system. So that's a drawback. There have been some efforts to make almost a human on a chip by looping several of these different organ tissues together. It's very challenging because we've found that we can't really replicate the blood quite as well. So one cell type might need a certain type of media, and the other type has another cell type has different requirements. So you've got to balance that. Also, cells in vitro grow at different rates, so that needs to be balanced as well. And then a lot of these chips are coming from the liver then to other organs because the liver metabolizes drugs, and we want to look at what the effects of those metabolites are on the other systems. There's differences in the in vitro flow rates and exposure in that respect. So some progress has been made. There are some of these that are very useful, but there are some limits to using them. And it's kind of a busy slide. I just wanted to point out here that some of the advances where it's contributed to drug development. And if you look at the highlighted parts, in the first one, they've used it for cardiac safety. Another one has been developed for liver. Another one is the airway on a chip. And what I'm trying to drive home is that we've made advances, but they're in organ-specific areas. And these chips are very useful when we take them in sort of isolation and look at the organs as well. So we're making our way up to the top here. But so we need to look sort of at the toxicology approach with this a little bit differently. So I mentioned it, toxicology is pretty much a fishing approach. When we look at this and we have tissues on a chip that are very organ specific, and the term organotypic has been used for this. If we know a drug has a certain toxin to a certain area, we can perhaps use that in vitro technique. It's more of a targeted approach for a specific organ as opposed to the fishing expedition. And once we use these chips, we can actually reduce animal use. However, sometimes that even may need to feed back into the toxicology studies as well. So I'm going to provide some examples of this to hopefully provide some clarity on this diagram. So my first example The organ that we're going to look at is bone marrow, and the fit for purpose. So we have to choose the type of chip that we want to use to answer a specific question. And the fit for purpose, or the question that we're going to ask, we're going to focus on toxicity to bone marrow cells. So I made up a company called OncoX, and they've generated a chemotherapeutic, and they're putting this into the clinic. And what they're finding is that it has severe bone marrow toxicity, that the patients just can't tolerate it. They develop certain toxicities. So they go back to the chemist at OncoX, and the chemist has said, I've got 4 other derivatives. I've moved a methyl group over here, a hydroxy group over here. I think these compounds will be less toxic than our first generation compound. So let's put these in rats and see what happens. So in this hypothetical design, what we're going to do is we're going to have to try it against our clinical drug in the animal. We'll need negative controls and 4 new candidates. So if you do the design, it's going to take 30 rats to test, at least do the pilot on these 4 different compounds. But another group that's working on in vitro systems says, "Oh, hold on here. I've got an in vitro system that tests the bone marrow, and this is called the colony forming assay. It's much like a bacteria plate, except we're going to plate bone marrow cells on there. The bone marrow cells are going to grow up and form colonies. So when you expose these colonies to different toxins, you can determine how toxic they are by counting the colonies. And you do this with increasing doses. So let's go back to that diagram there, looking at therapeutic indexes. This is one experiment. I've separated it for clarity on 2 different graphs. So the first one, the upper graph, looks at the results of our controls. And let's say, for the sake of the argument, the efficacy of all of these drugs is the same. It's between low and medium. So that's indicated by the green arrow. We add a positive control that we know that is bone marrow toxic. We want to make sure our system's working. So you see that over in the far left there. And yeah, we don't have to add too much drug to it before it kills all of the cells. And then our first generation compound, we see it confirms. Yeah, it's pretty close to that therapeutic index. But if we also include the negative control, we have to go out quite high before we start killing cells. So we know our system is working. In the same experiment, though, we've ran our experimental objectives. And so here we look again, and we see that our comparing it to our candidates, number 3 is even closer. So that's a pretty bad player. Number 2, not a very high dose. It causes a lot of toxicity. But look out here, we've got number 2 and 4. They're much better than our first generation, and they don't kill the cells that much. So what the company then decides is, well, 1 and 3 in this in vitro assay look like they're pretty bad players, and we're going to use this bone marrow assay to predict it. And we're only going to take compounds 2 and 4 together. We've actually saved 10 rats that we need to drive forward. So it's a reduction. We've used the MPS in a reduction. Another example, and this is partially from mine, I made up part of it, and part of it is true. So the first part, we're developing nanoparticles that target the lungs, and we put fluorescent dyes in them. And you can see over in the schematic from our publication that the lungs light up with 2 of our candidates here. So we think we have a great method of developing drugs specifically to the lungs. But we need to hold on here. We've run this study in mice, and we look through the literature, and we find that the mouse is different than the humans in their physiology. So what we can do is we can develop with some further, this is all hypothetical, for some further testing. Perhaps maybe we can use the mouse lung chip and compare it to the human chip, run our nanoparticles on both of those to see if we can replicate those to determine if the mouse is accurate in predicting it. So what we'd be looking at is similar results on the mouse lung chip versus the human lung chip. And that can help build confidence as we drive our drugs forward. We can do the opposite with that too as well. Sometimes we'll see what we call exaggerated toxicity in the animal model, and we may think that the animal has a different mechanism for digesting or working with that drug. We could run a lung chip and see if perhaps the human doesn't respond the same way. We might be building a case that, yeah, the animal model gave us a false lead. So another application of the MPS system. So coming back then, again, we're looking at these individual chips. They're organotypic. And perhaps we may get to some point where we run several different panels of organs at some point and demonstrate that they're safe. I don't know if we'll ever get to the point where we'll develop a panel of anywhere from 5 to 10 of these chips that will be more predictive than the mouse model. So it's one area to consider that we're going. But again, these chips are taken in isolation. They don't represent the whole interaction of the system that we see in the animal. So we can have some success, I think, but we have to put some disclaimers on this with using the MPS systems. It's got to be a targeted approach. We need to know what organs are affected. We have to have a specific question in mind, and then take the chip and fit it to our purpose. Find the chip that works, that will address our question, and we have to know the limitations of our chips. So I think, again, we can use the MPS systems and lead us to success, but we have to put some qualifiers behind it. So at this point, I want to provide just a little bit of my perspective. This is coming from my experience. It's not fact per se, but where I kind of see things going and some different aspects that we need to look at. So I think one of the things that we got carried away is we're looking at MPS systems as a route to reduce animal use. I think we ought to be looking at it at the different perspective. The introduction of MPS systems, I think, have made great strides in improving the science over our monoculture, tissue culture results. So I think we've made great advances, and we need to look at that end of it as how we've improved our in vitro technology. And I'm even willing to bet that in 5, maybe 10 years, we're going to be seeing more MPS systems being tested and used than we are the tissue cultures over on the left, where we take a single cell type and expose it to plastic. I think we get much better in vitro results. So I think we need to look over on that end. The other thing that sort of came to mind, and this goes back to when I was working in the academic lab, and I would, for a tumor model, I'd have to grow numerous cells in these tissue culture flasks, the T75s, T100s, and use these pipetters. I generated a ton of waste, and if you figure everybody in the lab, we're producing a ton of plastic biohazardous waste. Well, these are called microphysiological systems, and the amount of plastic that we use, and we can use pipettements instead of pipetters. I think we could potentially, in some cases, maybe not all, really reduce the amount of plastics that we have coming out of our labs as we adapt this technology. So it's just another way of looking at the microphysiological systems. So coming now down to the home stretch, are we going to phase out animal use? And this is sort of my perspective here. I think AI is going to have a bigger impact on reducing and replacing animal use. And the way that it's going to work is we're already making great strides in identifying how protein structures through modeling, how they fit into certain pockets, how they interact with receptors. So I think to start with, we're designing better drugs and making better candidates through AI. Now, what this AI then has to go back to is we need to kind of track our hypothesis with these structures and get feedback from the clinics. Is this really truly happening? Where we're predicting that we're, through the protein structures, making safer drugs. Did it really happen in the clinic? That information has to be fed back into the AI system so that we can test and make it better. And I think this is going to take anywhere from 10 to 20 years to get all of this data assimilated and put together. These hypotheses from the structural analysis, we're going to use animal models, we're going to use MPS and clinical studies to feed back into these systems to make better drugs. So MPS will play a role in it, but I think AI is going to be the main driver. In order for this to work, I think we need to have adequate communication. Again, AI needs data, good data to work. And so everybody needs access to this data so that we can munch away at this challenge. The other aspect, and I didn't touch upon this though in doing the research, clinical studies are not need some improvement as well in defining more predictive medicines, and there are some errors that are made. So the clinical studies are the benchmark. The clinicians need to work on this area to improve the clinical studies, because if we can't get sort of that metric on the clinical side right, we need to pair our lab work to those. The other thing that I think will impact this is societal shifts and how we view animal use. And what I mean by this is, there are efforts, and you've probably read about where they've produced meat in a lab and without using animals. And if they get this technology up to the point where it's palatable and nutritious, and let's say that we begin to shift from using agricultural animals to more technology that's developed meat and our food substances in the lab, and that becomes successful. If we start reducing the amount of animals that we use in agriculture, how is society then going to look at the use of animals in the laboratory? So there may be some pressure, and that bar for animal use may rise and lower depending upon where the technology takes this. I didn't mention it though, the in vitro systems are quite expensive, and so we've got to invest quite a bit, and that those resources need to be continued over the next 10, 15 years to make an impact. So again, I hope this wasn't too much of a downer. I think we're going in the right direction. We need to keep flying high and develop the technology and drive it forward. So our balloons are still high up in the air. And so I again encourage you to visit the 3RC Microphysiological Systems. They've got a lot more information. You can learn about it. Visit their technology hubs, see what it's all out there. Again, I'm open to questions. I'm starting a new biotech consulting firm that's coming out fairly soon, a new business. Also, you can link with me on LinkedIn. I'm happy to answer any questions or any follow-ups, and I'll turn it back over to Tom if there's any questions or comments, and thanks for hanging out with me.
Thank you, Dr. Peterson. Yes, there are a few questions that have come in. For those of you in the audience, do please feel free to submit your questions now, and we'll get to those in the queue. The first one here. Anti-lab animal groups have taken over the narrative and seem to position MPS as replacements that are readily available and currently more predictive, I assume, than lab animal research. How do we in the research field respond and take back the voice on these issues?
Yeah, no, definitely. We've been recognizing that over I would say the last 15 years, where they're playing more and more of a role. And I think we need to get out there and communicate the reality of it all. And we have organizations such as this one, the 3RC, and a variety of others that try to set the narrative straight on that. So I think it's a matter of getting out there, getting the communication. The challenge, I think, is always, and always has been, is that these special interest groups appeal to sort of the emotions. They can say a couple, make a couple comments that say, "Oh, and we've got technology. We don't need to use animals right now," and it gets people riled up. But for the scientific community to kind of counter that, just as I did today, we've got to explain things and sit down with people. So that puts us a little bit of an advantage, is that what we do is technically challenging, and we have to communicate that back to the public. But I think just supporting groups such as NJABR, the 3RC, to be our voice and get out there, I think is very important.
Thank you. You showed some slides representing MPS systems viewed more as a reduction type of technology. Do you see them being a viable replacement for the use of all animals in research?
Yeah, I think the start is, and at least in my experience, we've used them as a reduction, and I gave some of the examples there where you've got 5 candidates going forward, and if you know what the toxicity is, you can get rid of some of the bad players and use fewer animals. I think replacement might come in toxicology. There are some special areas that the FDA may ask, such as testing for cardiac toxicity. So instead of and usually a lot of these studies are run in parallel, but if you would have to go back and repeat a cardiac study, perhaps once we get these in vitro heart models developed and enough information comes through, perhaps at some point we can use that as a replacement for the cardiac testing. But again, we need sort of that feedback loop in saying, yeah, the in vitro tests show this. Did it really happen in vivo? Did it really happen in the clinics? And we constantly need that information before we can build confidence in those areas. So in some respects, it can be a replacement. I think we should think of it as reduction for the time being, because I think that's where the greater application is.
Thank you. There's 2 related questions, so I'll ask them together here. Are the data requested by the review team going to accept MPS? And do you see them referring to the review team as a roadblock to greater MPS integration and use?
I think it'll be up to the scientists to integrate it. And I always think of science and drug development as a puzzle. So your in vivo model alone isn't going to provide the definitive answer. You've got to provide some in vitro data as well, perhaps some flow cytometry. So we have a variety of tools in our arsenal that we use. And MPS is now being added as one of those tools to help put that puzzle together. And so when you think of applying for a certain a drug to the FDA, you're actually handing over a package to them. Think of it as a package with a variety of different components. And each of those components adds a little bit more confidence to what your drug is. And so the FDA reviewer will look at that and evaluate, well, do I have enough information? Are there enough pieces of that puzzle together to give a green light to advance that? Or they may come back and say, well, we're not quite sure about this result or that result, and they may ask you to repeat something or add additional data. So again, I think it's a new tool that we can use to improve our science, and that's what it's all about: is doing good science to make sure that our drugs are safe.
Thank you. the second part to that was, and I hope this isn't viewed as a political question. Do you think we need a strong mandate from the federal government to advance MPS and NAMs?
Yeah, well, I think it definitely helps, and you can see now that NIH has invested money and built up an infrastructure. We're also seeing some companies as well building up their MPS, so it definitely has a positive impact on the direction that we're going, and that's sort of why I had the balloons up in the air. I think this is great, for a person that's worked in the 3Rs arena for a long time. I think this is great. Where we have to be careful, though, is how the public is, as was alluded to in the prior question, how the public perceives this and the misinformation and expectations from the public. We need to be realistic about it. And hopefully the information I provided here helps us bring us back to that reality.
Thank you. What are your perspectives regarding animal-based NAMs versus human-based NAMs? The focus appears to be solely human-based. What are your thoughts? Are animal-based MPS relevant?
Yeah, so I gave the one example, and I think it's the value of the animal-based studies. Well, first of all, if for example you see lung toxicity and you have a clue that perhaps there might be something different between the animal and the human, you can then run in parallel the human chip against the animal chip to determine if that mechanism is still present. Perhaps you've got exaggerated toxicology in the animal model that you don't see in the human. So you can use that to kind of diffuse it and add information to it. I think the FDA reviewers would still be somewhat skeptical, but I think it helps if you have other evidence. It can help sway them. So I think it's what we call comparative medicine on an in vitro level. The other area that animal chips come in, and we've done this before, the human cells are very expensive to get from the suppliers. And if you can develop your own animal chips, you can actually use the animal chips to validate your platform. And once you get it going with the animal chip, you kind of know where you're going to need to troubleshoot and use that as a lower expense mechanism to make sure your platform's working right. And then you can go then to the human chips, the more expensive model with higher confidence.
Thank you. I think this is a good, more practical question here that just came in, and I think it speaks to your career starting as an AV and then moving to integrate more of these technologies. Do you have any recommendations for skill sets a strictly in vivo worker should look to build in their repertoire for this somewhat changing job landscape?
Yeah, I always think, for research scientists, lab animal veterinarians, I always think it's a great idea to have sort of a subspecialty. I think it really helps in your marketability. And I've worked with colleagues like in the lab animal field that are really expert surgeons. And so they still do their lab animal jobs, but they have this expertise of surgery that they can develop. Ophthalmology, the same. I once worked with a lab animal vet that really liked the ophthalmology area. Toxicology is another area that you can do in together. So I think it counts also for researchers. It's part of the reason. At least when I went through the programs, it was always a good idea. You did your graduate work, your PhD in one area. Now, if you want to do a postdoc, do it in something that's somewhat related but different, just to give you that variety of experience. And I think it just makes your marketability so much so much better. So Um, yeah, I think, again, I'm looking in my crystal ball, maybe 15, 20 years from now, uh, uh, we're going to start making that transition. So it's a good idea to have some experience in cellular and molecular biology. It's kind of where my interests have gone to. So the other aspect is don't force yourself to go into a specific area. Follow your passions. But again, I've always believed in being flexible. Uh, when I first started out, I told people I want to do lab animal medicine, and I also want to be a researcher. They said, no, you can't do that. You got to focus on one or the other. I kind of ignored them. I think the flexibility aspect kind of delayed me a bit because I didn't focus in certain areas. But I've had a lot of fun doing it because you're not stuck in one area at one time. So, uh, short answer is follow your passion.
Thank you. Um, this is a long one here. So your slides um, earlier in the presentation referenced the low numbers of adverse outcomes in preclinical, I guess, data, preclinical um, data leading to few adverse outcomes for humans in some of the clinical trials. What can be done, and what recommendations may you offer to assure the public that we will continue to have uh, safe data and ensure that we don't have adverse outcomes with less reliance on lab animals? Hope that makes sense. It was a long one. I tried to
Yeah,
combine it for you.
yeah. Again, doing good science and building confidence, building translational models. The MPS is going to be part of that. And then, as I mentioned at the end, the AI to constantly feedback and grow on the information that we have. Clinical studies also need to be improved as well. We're getting into an area called predictive medicine. And so for a long time, we've taken, for example, a chemotherapeutic and put it into anybody that had a particular cancer. Now we're getting to the area where we're looking at a particular drug that targets a specific protein. We'll analyze the patient beforehand and say, is that protein elevated, to hedge our bets that that drug is going to attack that protein. So our clinical sciences are getting much better as well, and we're improving in those areas. So using the science to improve our predictability, and that's a challenge. The big word is predictability, and I think it's the holy grail that we're going to be constantly working on, and using microphysiological systems, AI, and better models, and improving our clinics. I think will raise those success rates much higher.
Thank you. I have one final question in the queue here. Do MPS have more short-term value and impact? Well, I misunderstood this one. It appears that MPS have more short-term value and impact on the safety and toxicity issues. Do you see them having being able to have long-term impacts on the efficacy side of the issue as well?
Yeah, so I think it just depends upon what disease you're talking about, and I guess it goes back to one of the challenges I think that we have with MPS systems is doing sort of in toxicology. Once you do your single dose toxicology study, you've got to go to a multiple dose toxicology study. How do you replicate that in an in vitro system? That's one challenge. So I guess the question is sort of on chronic types of diseases. And I think, and I've always sort of thought, we can't be looking at diseases as a whole type of disease. We need to look at the mechanism that's causing those diseases and that continually drives those diseases. And the MPS system, because it is a minimalist approach, we can look at the mechanisms that cause those diseases. So, for example, in asthma or some certain lung diseases, fibrosis causes lung impairment or breathing impairments, and that can be a chronic disease. If we can identify sort of the mechanism that's causing that and test that in an in vitro system and stop that mechanism, then I think you have application for both acute and chronic types of diseases. Hope that answered the question. If not, I'll be happy to follow up with the person that asked it.
Thank you. So that brings me to the end of the questions in my queue. I just wanted to thank you for taking the time to present here. We did have a number of comments come in. Thanking you for the great presentation. So I just want to echo that. We appreciate you and the participants as well for taking the time out of your schedules to be here. I'll just kick it over here, and so I just want to conclude and thank Dr. Peterson for your time again. Another wonderful presentation on the topic. So thank you for offering double duty here with the conference and our webinar series. It is very much appreciated. And as always, thanks to the 3Rs Collaborative for their ongoing partnership and collaboration. No pun intended there with our efforts to provide some 3Rs content for the community. So I hope everyone enjoyed this, and we will see you again next week. Thanks, everyone. Have a great day.